Other than diffusion of gases, we also have diffusion of solutes. Solute diffusion is important for nutrient uptake. Nutrients like glucose, amino acids, and ions can diffuse into cells, where they can be used for growth, repair, and maintenance of the cell. When the cell has used these nutrients, sometimes we have waste products being created. These waste products need to be removed from the cell. Diffusion is critical in waste removal because it allows products like urea to diffuse out of the cells. The importance of this is that it prevents toxicity. When the particles moving in diffusion are water particles, the diffusion is given a special name, which is called osmosis. So water diffuses through partially permeable membranes by osmosis. You need to know how to define osmosis. Osmosis can be defined as the net movement of water molecules from a region of higher water potential, which is a dilute solution, to a region of lower water potential, which is a concentrated solution. This is done through a partially permeable membrane. So this definition is very, very important. You need to make sure that you remember this definition. So water can move in and out of cells by osmosis through the cell membrane. Water also enters the roots by osmosis. Another type of movement into and out of cells is active transport. Examiners like to compare active transport to diffusion. So I'm going to define active transport so that you may be able to differentiate between active transport and diffusion. Active transport is the net movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration. This happens against a concentration gradient, and it uses energy obtained from respiration. So as you can see, there are some similarities between active transport and diffusion, in the sense that both talk about the net movement of particles. However, unlike diffusion, active transport talks about these particles moving from a region of lower concentration to a region of higher concentration. Diffusion talks about these particles moving from a region of higher concentration to a region of lower concentration. Diffusion also talks about the particles moving down a concentration gradient, whilst activation, or active transport rather, talks about the movement of particles against a concentration gradient. So unlike diffusion, active transport also uses energy from respiration. So please make sure that you're able to define active transport, and you're also able to compare active transport against diffusion. Now, the importance of active transport is that it allows cells to selectively take up specific ions or molecules to maintain optimal concentrations of essential substances within that cell. So if you take a look at a plant which is growing in soil which is not very fertile, that plant is still able to extract mineral salts from the soil through active transport, is still able to absorb nutrients from the soil through active transport. It does this by using energy from respiration, and it will be moving these nutrients from a region where they have a lower concentration to a region where they have a higher concentration. So these are the aspects that define active transport, and these aspects need to be remembered. To highlight the key points, diffusion talks about the movement, the net movement rather, of particles from a region of their higher concentration to a region of their lower concentration down a concentration gradient. Osmosis talks about the movement of water or to be precise, the net movement of water across a partially permeable membrane from a region of higher water potential to a region of lower water potential. Active transport talks about the movement of particles from a region of lower concentration to a region of higher concentration through a partially permeable membrane. And this process here, unlike diffusion, requires energy. Now, factors which affect diffusion include surface area, temperature, concentration gradient, and distance. With regards to surface area, smaller particles or particles with a smaller surface area, you've got a faster rate of diffusion. Higher temperature also increases the rate of diffusion. A steeper concentration gradient increases the rate of diffusion. A shorter distance increases the rate of diffusion. So we're saying that if the distance that the particle needs to move is short, then the particle is going to move from point A to point B in a very small amount of time. Now, there are some terms that we need to also familiarize ourselves with. This include turgid, plasmolysed, and flaccid. We will be using those terms in this slide. Now, to establish how these terms are used, let's take a look at this scenario whereby we have a potato strip which is placed in a solution. If the solution has a lower concentration of solutes than the potato cells, water will flow into the potato cells. This will cause them to become turgid, which means swollen. And the reason why the potato cells become turgid is due to increased turgor pressure. The potato strip may become firmer and its mass may increase. On the other hand, if the solution has the same concentration of solutes as the potato cells, there will be no net movement of water. The potato strips will remain relatively stable. They will remain the same as they were before. If the solution has a higher concentration of solutes than the potato cells, water will flow out of the potato cells, causing a decrease in turgor pressure. The cells would become flaccid, which means that they'll become soft and limp. The potato strip will lose some mass. In extreme cases of high solute concentration, the potato cells may undergo something called plasmolysis. This is a situation where the cytoplasm shrinks away from the cell wall due to severe water loss. We are done with topic 3. We're now moving on to topic 4, which is biological molecules. So the macromolecules that we need to learn about are carbohydrates, fats, and proteins. Carbohydrates and fats are made from the elements carbon, hydrogen, and oxygen. Proteins are also made from carbon, hydrogen, and oxygen, but in addition to these three, we also have nitrogen. Macromolecules are large molecules like starch and proteins. These macromolecules are made from smaller molecules. For example, glucose, a smaller molecule, is used to make starch. Amino acid, a smaller molecule, is used to make proteins. We also need to know about the tests which can be carried out on different types of molecules and different types of foods. So we've got the iodine test for starch, Benedict's test for sugars, Biuret test for proteins, and the ethanol emulsion test for fats. We will take a look at those tests shortly. Before that, we need to remember that large molecules, like I highlighted earlier on, are made from smaller molecules. So some examples of large molecules, these large molecules are also known as macromolecules. Examples of these macromolecules include starch, glycogen, and cellulose. These three are all made from glucose. Glycogen is found in animals. Starch and cellulose are found in plants. We also have another macromolecule which is known as protein. Protein is made from smaller molecules called amino acids. Other macromolecules include fats and oils. These are made from fatty acids and glycerol. You are expected to know about the procedure and the expected results when you test for certain substances in biology. Now, the first substance that we want to test for is starch. So to test for starch, we use iodine solution. The procedure is to add iodine solution to the sample containing starch. After this, you should observe the color change. If you get a blue-black color, it means that starch is present. You can also test for reducing sugars using Benedict solution. The procedure is to heat the sample with Benedict solution. After this, you observe the color change. If the color changes from blue to any one of these colors, green, yellow, orange, or brick red, then it means that reducing sugar is present. Now, the color also determines the concentration of the reducing sugar, with brick red indicating the highest concentration of reducing sugar, followed by orange, then yellow, then green. So green indicates the least reducing sugar. If the test just gives you a blue color, then that means that there is no reducing sugar present. Another test that we need to be familiar with is the test for proteins. To test for proteins, we use the Burette test. The procedure is to add Burette reagent, which is actually copper 2 sulfate, to the sample that you are testing. After this, you need to observe the color change. If you get a purple color, it means that proteins are present. Last but not least, we want to look at the test for fats. To test for fats, we use the ethanol emulsion test. The procedure is to add ethanol to the sample and then shake. What we expect is for the fats to dissolve in the ethanol. After this, you now add water. Fats do not dissolve in water. So when you add the fats to water and shake, you should observe an emulsion formation. The expected positive result for this test is a milky white emulsion which forms, and this milky white emulsion indicates the presence of fats in the sample that you are testing.